Hello, today I'm going to talk about a web-based program that is freely available from the UC Berkeley site, and it's called a Tinkercell. It's a computer-aided design for synthetic biology. This is the main page of Tinkercell. I'll show you how to download. Click the download. There's a Windows installer. Click it, and there would be instructions to download it. This program is basically used for designing regulatory networks inside cells, and how they would act. You can actually simulate them, see the kinetics of it, the stochastic modeling of it, and then decide whether your circuits are going to work inside a cell. There are various tutorials which you can see to get used to it. There is a slideshow which you can click and see what's inside. It will so show you that databases, computer-aided designs, how the interface looks like, what are the components of it, how we can do the mathematical modeling, then how it can be run, and all sorts of things which would give you an idea about whether your circuit that you designed is going to work properly inside your cell. What is the consequences of running that program inside your cell of interest. So it's basically based on 3 programming languages, that is C, Python, and Octave. Now, and there are various blogs for this, and you can say what you want, or you can actually help them improve it, because it's an open source software. Let's go to the Tinkercell. There are tutorials which you can learn from the website. This is the Tinkercell interface, and we would work on Tinkercell. Let me show you what are the parts available here. There are different molecules that we can select: transcription factor, enzymes, proteins, RNAs, reporter. There are parts that are useful for designing our circuits: compartments in cell or subcellular compartment, regulation, how it can be regulated, like is it an enzymatic catalysis or an activation reaction or a repression reaction. We can design anything. And there are reactions, from which direction one reaction goes to where, we can find out. There are inputs, various inputs, wave inputs, and that is necessary at times. And there are various modules. If you need, you can change how it looks, how many parts, modules, and compartments they show. You just have to click all the parts in the catalog and then run your program. So let's design what we want, and our engineered Ribo-regulator. I'm going to show you today how to design an engineered Ribo-regulator that would give rise to a cis-repressed RNA, which represses the protein production, GFP protein production, inside a prokaryotic cell, and it can be transactivated by another set of promoter, that is the transactivating RNA. The cis-activating RNA and transactivating RNA binds together. There is a conformational shift which allows the cis repression to be, which allows activation, and this cis repression would be deleted. And how this happens is a protein that gets produced from the prokaryotic gene, when the protein is produced, we can say that it has been transactivated, and if the GFP doesn't, if we cannot see the presence of GFP inside the prokaryotic cell, we can say that it has been repressed. So let us first make the parts. What parts we need for for all the protein coding regions? We need a promoter. We would need 3 promoters. These are the 3 promoters, coding sequences for 3 of them. These are absolutely necessary and you can't live help without them. Ribosome binding site, terminator, we would need some parts. This is for GFP, and that's pretty much it. And let's assemble this promoter, RBS. I'm going to pause this video and then continue after I design the parts, and you can, that way I can save the time. So this is how our system looks. I take this GFP RNA, we can name them as in however we want. So this is pretty much the reaction that we want, and we can simulate this inside the cell analysis, flux based balance analysis, or we can simulate deterministic simulation. Let's go with this default thing. I don't know, it's saying that there's a system, there's some syntax error. I am not pretty sure of what this means, but these are the systems that are there. I can just explain you how it works. This is the cell inside, this is the prokaryotic cell inside which there is a promoter, there is a ribosome binding site, and a gene sequence and a GFP that is tagged to a termination sequence. The protein, the mRNA is synthesized, and from the mRNA the GFP protein is made. We can change the GFP protein if we want, I guess. Replace, okay, sorry, it should be replace. Okay, replace graphics. You can find something suitable like a reporter and replace. So this is a green fluorescent protein that is being made inside the prokaryotic cell. We can repress this with the help of cis repression RNA, and when the cis repression RNA binds to the transactivating RNA which is produced from another promoter, there is a conformational change, and this again activates the production of the protein or the GFP. So this is an example of an engineered Ribo-regulator, which has already been designed. I just showed you how to do this and simulate inside a cell. I wish this worked. I'd show you how this works inside the cell in another tutorial probably. See you for now, and I hope you enjoyed.